So good morning, everyone, and thank you for attending this first early session of the very first day. And it's really my pleasure to chair this, multimodal session, I'd say. I had a co-chair with Risto Ilmonyami, who couldn't make it to be here, but we conceived like this session thinking about what like other modalities like fMRI or DTI or EEG, as in my own case, can like support TMS for targeting of the stimulation. And also we have two speakers, which are Robin and Sean, which will also introduce how these kind of approaches can help to optimize treatment and in different kind of patients. So one thing that I have to mention is that unfortunately Baran had the last minute issue, so he sends you his greetings from the hospital. Nothing really bad, but anyway, this prevented him from being here in person. And so for this reason, we will start with this video recorded lecture, but we are lucky to have Victor Sousa, who is also sitting there next to me, who is one of the authors of this work, and we'll be happy to take the questions and to further discuss the topics of the first talk with you. So please, can you send Baran's talk? Thanks. Yeah.
The streamline, and the set of the streamlines is called a tractogram. Here the color coding is made based on the seating location, that is the starting point of the streamline on thalamus in this case. But what is real-time tractography guided TMS? I think this video explains it the best. What we see here is an operator holding a TMS coil. The neuronavigation system aligns the coil and the subject's MRI using infrared markers placed on them. And we have a visualization system that shows the operator the field surface of the brain. Conventional neuronavigation systems can already do this much. But as additional information, we now also show the streamlines obtained with diffusion MRI-based tractography in real time, based on the coil position. I will next try to explain how tractography is done in general, and then how we do it in real time. We start with diffusion MRI data, which looks like this. It is a four-dimensional data. The fourth dimension contains information about diffusion for each voxel in the image. We first pre-process the data and then apply a diffusion model, which provides us a new image that looks like this. This is called the fiber orientation distribution, or shortly FOD image. The color coding here shows the direction of diffusion. Red for left-right, blue for inferior-superior, green for anterior-posterior directions. And the size of these glyphs show the strength of diffusion. Basically, we have information about the direction and strength of diffusion in each voxel. Then we pick a random point in the brain, which is called the seed, and ask a tractography algorithm to find where it connects to. And the algorithm step by step propagates in the FOD image to find the connection. Basically, we only need this FOD image for tractography, which does not change in the conventional or real-time case. What we do, of course, is to do the few hours long pre-processing and the diffusion modeling part in advance. So we only do the tractography part during real-time operation, which takes milliseconds for one streamline. In short, we use exactly the same tractography methodology and algorithm in real-time tractography, and there is no compromise made to gain speed. But is tractography reflecting the structural connectivity in the brain accurately?
An important effort to mention here is the ISMRM 2015 Tractography Challenge. Here, the organizers started with a ground truth tractogram, which was basically a combination of 25 major fiber bundles in the brain. From this ground truth, they simulated diffusion MRI data, which was given to the contestants. And the challenge was, of course, to reconstruct the connections as accurately as possible. The results show the following. Notice here that the x-axis refers to the non-existing or false positive connections, whereas the y-axis is related to the missing or false negative connections. So on average, on individual fiber bundle level, large portions of the bundles were found to be missing. And when they looked at the number of fiber bundle clusters, many groups submitted results that had far bigger than 25 bundles, which were present in the ground truth. The conclusion is tractography has big limitations and there is need to do better. To address this need, we have actually been working on our own tractography algorithm during the past few years. We are using our own parallel transport tractography algorithm, or shortly PTT. We have an open source software called Tracker that implements PTT. It's a command line tractography tool. It's written in C, C++. It has a Python wrapper. It has multi-threading features. It has no dependencies, so runs on a computer cluster quite easily. It has a website with full documentation, and the link is here. Of course, the main purpose of a tractography algorithm is to get accurate representation of structural connections in the brain. The goal is not to get them as fast as possible. And the strength of our algorithm is actually not about its speed, it's about its accuracy. And it has been winning awards in all tractography competitions since 2017. In 2021, we won the MIGCAI Tractography Challenge with Tracker, and also second and third ranking teams use Tracker as well. I have been working on Tracker since 2015, and this is actually what makes real-time tractography possible in our case, because this application is largely a software project. In Python, what we do is to import a tracker package, initialize it with one line with the FOD image, input seed coordinates, and run. The seed here is a basic NumPy array. That's basically all in its core. For real-time tractography, what we do is to set the seed points based on the information received from the neural navigation system. Basically, we continuously receive new seed points based on the coil position, obtain connections, and visualize in an iterative fashion.
And we are able to do this because we also have our own open source neural navigation software called InVisalius, developed by our team member Victor Souza. With our setup, thousands of streamlines can be visualized in a few seconds. Importantly, the results are not worse than what one can obtain with state-of-the-art offline tractography, because the same FOD is used. An algorithm is also identical. Plus, we include all other state-of-the-art tractography practices, such as the use of anatomical constraints. Furthermore, it turns out that real-time tractography has some interesting advantages over its offline counterpart. Firstly, it allows to tune the tractography parameters, which is highly useful in many applications. Secondly, we have a dynamic output, which opens up new opportunities for tractogram visualization. For example, in this case, what we show a concept called the uncertainty visualization, where more transparency is used to show those streamlines that are more likely to be present in the brain, unpresent in the brain.
I would like to spend a minute to explain the simple idea behind uncertainty visualization. As I showed for the result of the ISMRM 2015 challenge, it is possible to study false positives and false negatives in tractograms. And it is well known that there is actually a simple way to obtain results that would lay on a band such as the one shown here. The way to do it is simply to sweep tractography parameters. In this case, the FOD threshold parameter, or min FODM in tracker jargon. When we have a large threshold, we know that we will have less false positives.
But when we decrease the threshold, then we will have less false negatives. Real-time tractography gives us an opportunity here, where we can in real time sweep through this parameter and visualize those streamlines obtained with lower FOD thresholds with higher transparency. The results look like this. If we combine all the tractograms without uncertainty visualization, we will obtain this. But if we use uncertainty visualization, we can put on with a much richer information about structural connectivity. It is important to highlight the challenges and promises involved with real-time tractography-based neuronavigation. Well, the main challenge is actually involved with conventional tractography itself. Tractography is well known to be limited and it cannot be blindly trusted. When we look at how well tractography matches tracer injections, which is the gold standard, invasive approach for obtaining structural connections, we observe that tractography misses large extent of bundles. Many times half or more of the bundle is completely missed by tractography. At the same time, when we look at end-to-end connectivity of large brain regions, then we observe that many times streamlines go and end in a completely wrong part of the brain. These are known problems of tractography which we are continuously addressing to improve. If you are curious about where tracker performs in the ISMR 2015 challenge, it's about in this band. So we still have work to do. But because we do not have a bulletproof formula for tractography yet, we need to augment it. So what's augmentation? Augmentation here means that we need to complement the structural connectivity information that we are observing on the display. The most accepted approach is to use prior neuroanatomical information, which means the operator needs to know what they are looking after. That is a fiber bundle of interest in real-time tractography. Another approach is to use electrophysiological recordings. TMS-EEG in this respect is excellent approach that creates a synergy with real-time tractography, which actually opens up new research opportunities. Real-time connectivity information is new. It provides additional information. So we used to look at these when doing neuronavigation. Now we are looking at these. Of course, once you start looking at a neuronavigation interface with real-time tractography, believe me, you will not want to go back. Of course, this also offers new opportunities for both clinical applications and brain research. But wasn't this trivial to begin with? Tractography has been around for 2 decades, so one can wonder what makes real-time tractography possible now? What changed? Does it require the latest and fastest hardware, GPU, CUDA coding, etc.? The answer to that is no. The computation is done in CPU, no CUDA is used. The hardware requirement is quite easily affordable and it's actually not steep. Our tools work in standard laptop workstations. Then does it have to do with the algorithm, or is tracker highly optimized somehow compared to the previous fiber tracking code? The answer to that is also no. Actually, the computational complexity of our algorithm is quite high compared to many other algorithms, and then this makes it slower. On the other hand, the tracker has increased accuracy, which is something we are willing to compromise for speed. There are actually CUDA-based fiber trackers that are maybe 100 to 1,000 times faster than tracker. The natural follow-up question is then, why was this not done before? The answer is this was actually done before. In 2011, in Slicer 3D, for example, a real-time tractography-based application was developed for neurosurgical planning. And in 2014, a software called Fiber Navigator was modified to have real-time capabilities, where one can modify tractography parameters on the fly. So the real-time tractography idea is not really new, it's quite old. But that was also not the main problem that we sat down to solve. Of course, one has to have an accurate and fast fiber tracker. It is just a prerequisite. But our main problem is, what's a good way to convey structural connectivity information to the TMS operator so it is most useful for brain stimulation? So what distinguishes our work is mostly the design and user experience. The displaying of tractograms on the peeled brain surface, which is one of the most conventional approaches for TMS, is a natural choice for us. That was not demonstrated before. The dynamic visualization of streamlines, as we do in our work, distributes the complicated connectivity information a long time for the operator to digest it. So unlike previous works, we are showing a movie, not a photograph. The uncertainty visualization is purely made for real-time experience, which is the first time transparency is used to convey information about connectivity. Overall, we do believe we have a unique and working solution for TMS neuronavigation. With that, I would like to end my presentation by thanking the funding agencies and my collaborators who supported this work over the years, and also thank you for listening.
So I thank Baran. Oops. Can you hear me? Yes. For this presentation, and we decided to take the most of the question at the end of the session, but maybe just one question, if there is any, we can take it now. Or you just like continue thinking on what you have seen, put everything together, and we discuss everything at the end. Okay.
Okay, so it's my pleasure to introduce Professor Laura Marzetti. She's a professor of applied physics in the University of Chieti, Pescara, and her main expertise in this is in developing methods for estimating connectivity in from non-invasive measurements of the brain activity. So thank you for organizing the session, Laura, and yeah, the floor is yours.
My pleasure. My pleasure to have all this group of great speakers and to also introduce my own work in this field. As Victor said, I came to this connection to TMS quite recently in the framework of a European project in which Baran, myself, and Victor are involved, which is an ERC Synergy grant to which we collaborate. But my background comes really from the study of functional connectivity, firstly with MEG and then with EEG. And even more specifically, I got interested in methods that assess functional connectivity from neuronal oscillation that you can derive from EEG or magnetoencephalographic signals. And that's under the hypothesis that this theory that has been put forward by Pascal Fries, but also had some roots in the past work of Wolfsinger and the people that leaded the field there, plays really, a crucial role in understanding how this coupling of neuronal oscillation works. Since the idea is that if you have three, let's say, neuronal groups, I don't have the pointer, but I think you can grasp that they are those circles over there. And if the respective neuronal oscillations are phase aligned, meaning they have some synchrony with some possible delays, like in the purple and in the blue case, then this allows somehow these two regions or neuronal pools to communicate. While if we are in the purple versus green case, in which the crest of the one oscillation is paired to the throat of the other oscillation, that this means that there are no chances for these two regions to interact. There has been quite a huge effort, I would say at least in the last 20 years, in the field of non-invasive electrophysiology to develop methods that are able to assess whether two oscillations are phase aligned or not, and also with some delays or not. And overall, they all share the same background idea, which is what you can see depicted in this right part of the slide. So basically, if the two oscillations are phase aligned and you calculate the phase difference, which is what is plotted in the round circle over that, it's a kind of circular histogram, then if there is a phase alignment, you will see a preference of phase differences in this circular histogram. So meaning they are all aligned, in this case, around 130 degrees. On the other hand, if there is no communication, so if the phases are not locked, so there is no synchrony between the two regions, then you are likely to end up with a fully scattered like phase difference in this representation, and this would mean that there is no coupling in the two sources. So whatever, I mean, if you are familiar with the functional connectivity literature in EEG or MEG, you will understand that there is really a huge set of metrics that people have derived along the years, but most of them, at least the ones that deal with phase coherence, they all share this underlying idea. But they are not perfect, as always. And one of the issues that we have when using these metrics is that they are like the situation that I described, so they can catch a phase difference, a stable phase difference, also for a infinite, in theory, or for a quite long data, for quite long the observations that you have. But usually, and especially if you want to try to apply this methodology in real time so that we can also, as also Baran mentioned, use functional connectivity from EEG together with the other anatomical information to drive the TMS, then you are given with a small portion of data. And so you are basically looking at the whole story from a small window. And this might lead to a really bad issue, because if you have true phase coupling, as in the top row, then you will likely see it, and that's fine. But also if you have uncoupled sources, like in the bottom row, and you look at these uncoupled sources from a short epoch of data, like the gray one that you have, we have underlined there, then it's also likely that you will be tempted to assess some phase difference preference in this circle, which we know was not the case. So overall, for phase coupled, for true coupling, we are safe, but for uncoupled sources, we might really ending up in saying that something is connected while it is not. And of course, we don't want this, because especially if we use this in combination with TMS, we want to save our dose, so that we don't want to stimulate when it's not the case. So what we did as a first step was to say that you want to do this in real time, and you have your connectivity going up and down between two regions, your functional connectivity in that box-shaped way. Then you can take a bunch of metrics used in the field. I'm not going into the details, but I will be happy to do so later. And you can pick some window lengths through which to look at the connectivity, and assess whether, you end in this fake connectivity situation, or not. Overall, as a take-home message that we put forward in our recent paper, is that as long as the window is shorter than the real connectivity, then you can catch the true connectivity that is there, and you are also less prone to false positives in your data. So the idea is now that we want to use these to study, in combination with TMS, and we decided to look at the most common, I would say, use of TMS, of most historical, which is to stimulate, for instance, the motor cortex, and then look at the response, the peripheral response in terms of motor evoked potential. And what we wanted to do is to go a bit farther with respect to what is currently available in the literature, because so far there has been some important use of EEG to study the relationship between the state of the cortex, so the excitability, let's call it like this, of the cortex, and the amplitude of the motor evoked potential. Nevertheless, all these studies, and there are quite some, have looked only at local properties of the cortex, and specifically in this case, at local properties of the motor cortex. But what we wanted to do is to make a further step and to assess whether the connectivity, the functional connectivity of the motor cortex to the rest of the brain, so the corticocortical connectivity state of the brain, can influence the corticospinal excitability as measured by MEPs. And this, I mean, if we are able to do that, then in principle, if we want to achieve the situation in which we have larger MEPs, then we could stimulate when we have some specific connectivity as measured from real-time EEG functional connectivity, and then go and hit the cortex at that very right moment, not based on the EEG information from one location, but based on the EEG information of the coupling between different brain locations. Okay. But before going into that, we had to really investigate for the first time whether this EEG connectivity has any relation to the MEP sites. So we took some data collected in the framework of this Connect Brain project that I mentioned before, which is 8 subjects, very simple like protocol in which a TMS pulse is delivered to the motor cortex. And then the EMG is measured over 2 muscles in order to assess the motor evoked potential amplitude. The nice thing with this data is that for each subject there are quite a large number of repetitions, so it's about 1,000 trials for subjects. Okay, so we did some pre-processing, which is basically artifact removal, concentrating on the mu frequency, which has been said to be involved also in terms of single oscillations in the brain in this cortical excitability. And we further went to the so-called source space, meaning that we did not, as most of the studies did so far, so we did not use the EEG data as measured by the sensors, but we also applied an inverse method, so a strategy to derive the signals at the level of the cortex. And then we estimated functional connectivity using one of the metrics, of the phase coherence metrics that you have seen before. In the period of time that is, before the TMS. We really missed a pointer. So in the EEG data before the TMS pulse. This is also, you have seen the term, you have heard the term seed-based connectivity imbalance talk, meaning that we pick a region, which in this case is the motor cortex, and we look at the connectivity of these regions to all the other regions in the brain. And here you can see I lost a bit track of time, so if you guys over there tell me how it's going. So what we did was we seeded the left motor cortex and the frequency of the mu rhythm. We calculated the connectivity with all the rest, with all the other voxels or cortical nodes in the brain. And this is the average map of all the subjects. And I mean, I was quite happy with this map since this is EEG connectivity that is usually quite widespread. While in this case, we were nicely able to assess that the spot that you see over there, which is white, and is connected with the other spots that you see in red, which basically corresponds to the supplementary motor area and to the homologous primary motor region in the right hemisphere. Okay, the reason why you don't see anything red around the seed is because of the metric itself, so that by construction doesn't tell that the region is connected to itself. But you should interpret this as white being connected to the 2 reds over there. Okay, so nice. We have some background connectivity before the TMS pulse in the brain, which is quite consistent across our subjects. But we then want to understand whether this connectivity has anything to do with the corticospinal excitability. Okay, that's the measure. And so we took pair number 1, So this one, and then the second pair, and looked at all over our subjects at the modulation of MEP amplitude in the conditions in which the left M1 and the left SMA exhibit low or high functional connectivity. So it's basically a median split procedure at the level of each subject, and then we looked, the median split is done on the basis of functional connectivity between these two regions, and then we looked post hoc, I would say, at how the MEP behaves in these two conditions, whether there is a change in the MEP or not. And that's the case. The modulation is not, I mean, striking. It's about 4% modulation, but still is significant. And we have this modulation both for the connectivity between left M1 and left SMA, but we also have a smaller modulation, but still significant, between the two motor cortices. So coming from the field of brain network, what I told myself is, okay, but if these two phenomena are not independent, so this would mean that if I consider only the trials in which both SMA and right M1 are connected to left M1, then I should see a further increase in this modulation of MEPs. And that's basically the case. I mean, it's not really, again, it's not really striking, but it's significant. So if I consider the modulation of the motor network, so motor network high versus motor network low, so we see a modulation of MEP amplitude. And this is mostly true for all our subjects. At least there are no subjects in which the situation is completely reversed, so no subjects in which we have low MEP with high connectivity and vice versa. There are subjects in which, at least two, in which the situation is a bit flat, but for the most of them, you really can see this modulation at the individual level. Then we wanted to compare this approach. This is a bit weird because of the namings and so on, but I will make it straightforward. So we wanted to compare the way of assessing the modulation of MEP based on connectivity with what had been done so far in the literature. So in terms of either simply the phase of the oscillation at the motor cortex, or the constant model, which basically means that there is no modulation. So we ran a linear regression model in which we put all these variables, and what we understood from this is that in the most of our subjects, you have that the motor network connectivity really plays the major role in modulating the MEPs, and if you have to add a second factor on top of that, you have the phase of the oscillation at the local M1. So overall, it seems that the way in which we can explain the changes in the corticospinal excitabilities by considering the functional connectivity within the motor network, plus eventually some local properties of the motor cortex itself. Okay, as I mentioned, the idea is now to take a further step to calculate all this stuff in real time and to stimulate when we see the connectivity up within that motor cortex in such a way that we will then be able to assess the brain state in terms of connectivity and to target our our TMS pulses in a single location or possibly in multiple locations, as we are doing in cooperation with the Alto group, in which they are developing these instruments to stimulate different locations at the time. So in summary, even in EEG, I would say that even at single level, if you take the right steps and the right pipelines, you can assess motor network connectivity in the baseline activity, so intrinsic activity. And then we also have observed for the first time that these long-range properties of EEG signal, they do impact on the corticospinal excitability, so on this peripheral signal that we have taken as a reference. And therefore, our hope is that they can really be used to design protocols for real-time state-dependent stimulations based on EEG functional connectivity. And maybe also in combination with other properties that are currently used to this purpose. Yeah, of course. We tested only this showcase, so the motor network, also because we had a clear output in terms of the MEP signals. But what we really think, based on functional connectivity literature in EEG and MEG, is that this network effect can be observed in different subsystems, also those involved in high-order processing, and that in the long term, we could really use network properties of the brain to drive the TMS stimulation, also using the non-invasive electrophysiology approach. And yes, finally, I want to thank all the people in my lab, and mainly Vittorio Pizzella, who is also sitting here, and the guys working, and that's a bunch of people working in the Connect to Brain project that I also wish to thank.
Thank you, Laura. We have time for one question. I also realized that we actually have the app here, so there are plenty of questions already in the app.
Okay, yeah, right.
So we can do those in the end as well, but if anyone has one question now, you can say. If not, maybe I can just make one of the questions in the app here.
Okay, thanks.
It's a short one. So for the EEG connectivity window, what's the range for window? Is it sliding window?
Yeah, it's a sliding window, but the length of the window, we tested from, I would say, 250 milliseconds up to 1 second, and the best results overall we got with something like 500 milliseconds. So the best compromise.
Thank you.
Okay, thank you.
We can answer that question again.
Oops. And our next speaker is Robin Cash from the University of Melbourne, who will make a further step showing how all these ideas come together in personalized brain stimulation.
Cool. All right. Well, thank you. So I'm going to present a brief overview of personalized brain stimulation, focusing on a technique known as transcranial magnetic stimulation. And this approach aims to optimize TMS therapeutic outcomes by aligning brain stimulation targets with person-specific brain network architecture. So the most common therapeutic application of TMS is for treatment-resistant depression, where it is typically targeted to the dorsolateral prefrontal cortex. And over the course of several weeks of daily treatment, some people respond very well, but many do not. And overall, the response rate is around 35 to 50%. This is not bad, considering this is a treatment-resistant population, but nonetheless leaves a lot of room for improvement. And one basis for this variation may be clinical or demographic factors, although these have shown somewhat weak and inconsistent effects across studies. Another potential factor is suboptimal spatial targeting. However, over the past 2 decades, the optimal TMS target site has remained elusive. So a range of targeting methodologies have been explored, and you can see here that these have targeted quite different subregions of the dorsolateral prefrontal cortex. So without going into detail, recent evidence indicates that connectivity-based targeting strategies, and in particular, targeting strategies that are informed by an individual's brain connectivity, show the most robust relation to TMS therapeutic outcomes. So which brain connection should we target? Several studies have shown that TMS treatment outcome relates closely to functional connectivity, functional brain connectivity between the DLPFC stimulation site and a deeper brain region known as the subgenual cingulate cortex. So this provides an obvious opportunity to potentially improve treatment outcomes by prospectively selecting the DLPFC stimulation target based on optimal connectivity with the subgenual cingulate cortex. The easiest approach would be to identify a single one-size-fits-all DLPFC target site that should work for all individuals based on or defined by a group average map of SGC functional connectivity across the DLPFC. It is worth adding that the initial reason for focusing on the subgenual cingulate cortex is that this region was consistently implicated in antidepressant response across a range of different therapies. And on this basis, the subgenual cingulate cortex was also selected as the target region for deep brain stimulation. So a potential disadvantage of a one-site-fits-all targeting approach is that there is substantial inter-individual variation in brain network architecture, particularly within the prefrontal cortex, as highlighted here in hot colors, yellow and orange, corresponding to increased evolutionary development within this brain region. And these areas can vary to such an extent that individuals can be reliably identified from a large sample based on their functional connectivity fingerprint. Critically, a few studies have delivered TMS to a singular one-site-fits-all group target using neuronavigation, but these studies haven't shown any compelling improvement in treatment outcomes relative to conventional targeting. So together, these findings provide a strong rationale for personalization of the DLPFC target site based and defining this site according to connectivity with the subgenual cingulate cortex. But in order to translate this approach clinically, we first need to know whether precise personalized targeting is even feasible. So if personalized targeting based on brain connectivity is feasible, it should be possible to reliably identify personalized targets that remain near identical in position across two different brain scans. However, when we started this work, a paper had just been published using conventional methodology, indicating that targets differed by an average of 3.5 to 4 centimeters between two separate scans for a given individual, even when using the best quality scans from the Human Connectome project. In other words, personalized targets were not reproducible, and personalization did not appear to be feasible. So without going into detail, we developed computational personalization methodology building on previous work by Fox and colleagues from 2012. This ultimately enabled us to precisely and reliably pinpoint optimal personalized connectivity-based stimulation targets. We verified in 1,000 individuals from the Human Connectome project that the sites we identified varied by only 2 millimeters or a single brain voxel between scans repeated the next day or even 1 year apart. Importantly, these personalized targets were not only robust, but also differed substantially between individuals, demonstrating that our approach preserved inter-individual differences. So interestingly, we also had familial data from the Human Connectome project and found that targets were more similar between monozygotic twins compared to dizygotic twins, non-twin siblings, and unrelated individuals, suggesting that these targets are stable and under genetic control. So with this approach, we see an increase in the reproducibility of personalized coordinates. We see individual-specific rather than generic coordinates, and we see a striking increase in the spatial correlation of the SGC functional connectivity map across the DLPFC region when compared across two separate scans. So for a 15-minute scan, conventional methodology gives us a spatial correlation of 0.2, whereas with this approach, this increases to 0.9. We then tested whether personalized targeting is clinically meaningful in a sample of 26 individuals who had previously had a brain scan and who received TMS for depression and for whom the stimulation target and treatment outcome was known. We retrospectively computed the optimal personalized stimulation target from each individual's brain scan, And we anticipated that individuals whose actual stimulation target was by chance closer to their optimal stimulation target would have a better treatment outcome. So here you can see the scatter of optimal personalized stimulation target sites across this cohort. And here you can see that our data confirmed this hypothesis. This figure shows that individuals who serendipitously received TMS closer to their optimal stimulation site consistently had better treatment outcomes. So, moreover, these findings have recently been replicated by Shan Siddiqui and colleagues in an independent cohort. And in our paper, we also showed that there was no relation to TMS clinical outcome when we instead computed proximity to any of the one-size-fits-all group targets that I showed you earlier. In parallel, other groups have been working in this area, and there's now preliminary prospective evidence emerging that connectivity-based personalized targeting may enhance clinical outcomes across invasive and non-invasive brain stimulation modalities. So across 6 separate but small studies, including an RCT just published in the American Journal of Psychiatry, the average response rate is 87% and the average remission rate is around 65%. This is vastly higher than with conventional targeting, where the average response rate is 46% and the remission rate is 31%. These findings are, in my view, some of the most exciting in the field right now, and clearly warrant larger clinical trials. The importance of brain connectivity in mediating clinical outcomes is also becoming increasingly evident with another form of brain stimulation known as deep brain stimulation. So several sites around the world are now implementing this methodology, and in Australia the approach is now TGA approved, so the equivalent of the FDA. And my colleagues have also recently launched the first Australian clinic to offer personalised TMS. The next key question is whether the benefits of personalisation based on functional connectivity extend to other brain regions and TMS applications. For instance, previous work has shown that multi-day TMS to the parietal cortex can enhance associative memory performance, the parietal cortex being a key component of the cortical hippocampal memory network. And as you might have noticed before, the parietal cortex is another brain region that has high inter-individual variation in brain connectivity. So without going into detail, we retrospectively analysed an existing data set where MRIs were acquired, and this multi-day TMS protocol was delivered. We knew the actual stimulation coordinates and applied our personalisation methodology to compute individual-specific optimised target coordinates. Notably, the original targets were personalised based on brain connectivity, but our approach offers some advantages, for example, a better signal-to-noise ratio compared to the conventional 3mm hippocampal seed. So these data showed once more that closer proximity to these optimal personalised targets was associated with better TMS-evoked improvements in memory performance, indicating that the benefits of personalised targeting appear to generalise beyond the prefrontal cortex, at least to the parietal cortex. In this slide, I wanted to quickly make the point that TMS tends to impact and modulate distributed brain networks rather than a single brain region, such as the local stimulation target site. So here, following stimulation of the cerebellum, you can see changes in brain connectivity occurring with real, but not sham TMS, across different regions in this cognitive network, but with no change in the control site. Other research has indicated that the most therapeutically effective TMS and DBS targets, as well as lesions eliciting symptoms, are functionally connected to the same disorder-specific brain circuits. And Shan's work has delineated a convergent brain circuit in depression. So this becomes relevant, as I'll show you in this slide. So a recent meta-analysis demonstrated that across the past 20 years of task-based imaging research into emotional and cognitive dysfunction in depression, comprising almost 100 neuroimaging studies, there was no convergence in terms of the anatomical loci that demonstrated aberrant activity during these tasks. This finding raised alarm bells in the field around the reproducibility of research findings. The lack of convergence might be partially attributed to the incentive for novel task designs across different studies, small sample sizes, and other factors. In my latest research, I considered whether these regions might be linked or functionally related from a connectomics point of view. And so I'll skip the details of the methods and null conditions, but in short, these divergent coordinates actually localized to statistically highly robust distributed brain networks. And importantly, these networks recapitulate clinically meaningful and independently derived models of depression circuitry. Moreover, when I used these networks as a template to define personalized TMS targets in the same way that I described earlier, TMS therapeutic outcome was dependent on how effectively this emotional dysfunction circuit was targeted. And finally, I just wanted to quickly thank Andrew Zaleski and those who contributed and shared data with us. And if you're interested in this research, yeah, please get in touch. I'd love to collaborate. All right, thank you very much.
Thank you, Robin. So any live question? Or shall we check?
Oh, we've got a question in the audience.
Cannot see it, but please go ahead.
Yeah.
Can you go to quickly to the mic? There are several, I mean. Thanks.
Hi. Great talk. Really cool stuff. Can this be applied? I guess this is a two-parter, but the first part is, can this be applied to other prefrontal cortex areas like the orbitofrontal cortex? And also, is the relationship with the subgenial cingulate mandatory for the outcomes, like the mandatory for the clinical outcomes?
Yeah, so yeah, our intention is definitely to apply this to other brain regions and psychiatric disorders, and essentially see if we can help to establish kind of neurobiologically informed stimulation targets, potentially personalized targets if that's warranted, if there's sufficient individual variation for a given connectivity pathway. And yeah, I wouldn't say that. Well, you can use any seed. It doesn't have to be the SGC. You can use an entire brain circuit, like maybe one of Sean's circuits as well. So, yeah, to define the stimulation target. Yep.
Thanks.
Yep.
So thank you very much again. And now it's Sean Siddiqui's turn, and Sean is working on similar topics at the MGH.
Well, thank you. So, I'm trying to go back. This is moving forward for some reason. Would you guys be able to go back to the beginning? There we go.
Yeah. Just that there is no pointer, which is...
Yeah. All right, there we go. So thank you for the introduction. Again, my name is Sean Siddiqui. I'm a neuropsychiatrist in Boston, based at Brigham and Women's Hospital, the new Center for Brain Circuit Therapeutics in Harvard Medical School. I'm here to talk about connectome-targeted modulation across symptoms and modalities. So recently, the question has come up sometimes of whether these sorts of techniques can be applied to other disorders and other brain regions. I don't know if you've heard this question, but we heard it about 2 minutes ago, right? So some quick disclosures. There we go. I've done some commercial, clinical, and educational consulting, but by far most of my funding comes from the National Institute of Mental Health, so I'm biased in favor of dimensional circuit-based models of mental illness. Now I should mention we're a little bit short on time. I think we're a little bit behind schedule, so I'm going to cut out all the bad jokes from this talk. I promise I'm going to leave the good jokes in, but I don't have time to talk about this one. This is going to take about 1 minute, and I need to cut a minute out of the talk, so I'm going to move on. So the clicker doesn't work very well, though, so I could have just told the joke. So this is mostly about targeting TMS and DBS. That's what we spend a lot of time talking about. That's what this symposium has been about. So currently, we often use scalp landmarks to identify a patient's TMS target. So in other words, we make somebody's thumb twitch by stimulating the motor cortex. We measure 5 or 6 centimeters in front of that and say that's probably the right spot to stimulate. So I'm going to say that again. We use a tape measure. The same thing that this guy's using to measure this banana is the same technology that we use to target electromagnetic brain stimulation. We should be able to do better than that, but we did it because these landmarks were based on specific things, based on locations of lesions that cause depression and stimulation sites that relieve depression. So it works. That's why we do it. But unfortunately, ever since then, we've been using the same landmarks to target all sorts of other things like PTSD, generalized anxiety, addiction, etc. Again, this clicker is not advancing. Could you guys help? Do you guys know why? There we go. And we had mixed results. It's not clear why. So some people question why, but I think the reason might be because we're targeting PTSD based on lesions that cause depression. Now, we have the ability to target very precise locations in the brain with TMS or DBS, but it's useless if we don't know where the right target should be. And so we need a reliable framework to translate lesions of TMS or DBS targets for different disorders. And we started to do this using data science in our lab, but we're starting to translate this into clinical trials soon. So let me talk a little bit about how we find the right spot. So traditionally, brain lesions have been used to identify the causes of neuropsychiatric symptoms. So let's say you take 3 different patients with 3 different strokes, and let's say they all develop the same symptom. Let's call it depression. We can look to see where those strokes intersect, and we say, there it is. That's the spot. That's where depression comes from in the brain. Now, of course, it's not always that simple. So over time, originally people started, this was sort of the predominant way of studying brain mapping. You'd look at the locations of lesions, locations of stimulation, what response that induced, and measure an outcome based on that. Now, as we all know, in the 1970s and 1980s, functional neuroimaging came around and started to replace this stuff. You could finally non-invasively study what would happen to a patient and do well-controlled experimental manipulation. But one of the unfortunate side effects of that was that stimulation and lesion studies started to go away. And this is what happened over the years. Stimulation eventually made a bit of a comeback, but we still often target stimulation studies based on correlative neuroimaging. And there's a problem when we started try to design treatments based on correlations. And let's take an example of a disorder like tuberculosis, which we know a lot about and we have good treatments for. So TB rates correlate with malaria rates, white blood cell counts, mycobacterial infection, and travel to endemic countries. But some of these things are useful, some are not. So for example, malaria rates are a spurious correlation. If you give anti-malarials, TB won't get better. White blood cell counts are reverse causal. If you try to reduce white blood cell counts, that'll actually make patients worse. You don't want to get rid of somebody's white blood cells. Mycobacterial infection, on the other hand, is a primary cause. We know that that actually works. Antimycobacterial drugs are curative, and the first ever randomized controlled trial actually already knew this before they started trying this as a clinical treatment. And then travel to endemic countries is a secondary cause. If you stay out of endemic countries, that will indeed reduce your risk of tuberculosis, even though that's not the primary cause of the disease. So in other words, to find treatment targets, we have to find circuits that are causally implicated in the symptom. It doesn't have to be the primary cause, it just has to be part of that causal chain. So in a perfect world, it looks like this, but in reality, it looks more like this. So you often have different lesions that can cause the same symptom. You look at them, some of them overlap, some of them don't, and people look at that and say, well, maybe stroke has some sort of relationship with depression, but it has nothing to do with the lesion location. And that's what the conclusion that we've come to for a long time. But in reality, sometimes if you look at the connectivity, you find an extra piece of information. Sometimes these things that don't overlap with each other might share some sort of common connectivity profile, whether that's structural connectivity or functional connectivity. They might still map to the same brain network. And we can hypothesize that stimulating that network might help to relieve that symptom. But that raised the question of, does that actually work? Is it a hypothesis, or is there any utility to that? So we started looking at this in a 2019 paper that you might have seen in Biopsych. where we found a human depression circuit based on focal brain lesions. Lesions that cause depression were more likely to be connected to that spot, the left DLPFC, which happens to be the spot that we also stimulate with TMS most of the time. So we said, oh, great, look, we have some sort of explanation for why TMS is working. If you break the circuit, it causes depression. If you stimulate the circuit, it relieves depression. But at the same time, Amit Etkin wrote this editorial in the same issue of the journal, where he kind of called us out. He said, it remains to be shown that lesion or stimulation anywhere within a particular resting state network has the same downstream effects on behavior or symptoms. It remains to be shown. Usually when they write an editorial, it's laudatory, but he called us out appropriately. He was right. And so that raises the question of how do you define causality in the human brain? This is an interesting scientific question, but it's also a clinical question. You need to know causal things if you want to start to manipulate them. So as a scientist and clinician, I think about this in both ways. And so before thinking about how to define causality in the brain, we have to think about how do you define causality in general. So you give an exposure, you measure the outcome, then you get in a time machine, you go back in time, you don't give the exposure, and you measure the outcome without the exposure. And of course, this experiment has been done before, but it requires a lot of power, like 1.2 gigawatts, and most labs don't have the resources to do all that. You have to harness lightning or do some Libyan terrorists. And I don't know, I know there are a lot of grad students in the audience, so you might be too young to remember Back to the Future. But I have to emphasize the Fox citation is not the same Fox that I usually cite. It's a different Michael Fox. So some of the people here might be too young to remember the original Michael Fox. But the solution to this is to estimate causality using natural experiments or prospective experiments, followed by randomized trials. We don't necessarily have to do the randomized trial first. For example, when we did a randomized trial for tuberculosis, the first ever randomized trial, they already knew the likely outcome because of mycobacterial disease. So we published this model for how to sort of estimate causality in human brain function earlier this year, if anybody's interested in looking at it more. But going back to the question of depression and lesions, so we wanted to really causally map depression, so we took all the different brain lesions that we could find, all the different TMS sites we could find, and like I said, we use a tape measure to identify our TMS target, so we get incidental variability in where anybody gets stimulated. So everybody gets a slightly different spot, so we can use that to our advantage. Same thing with DBS. The neurosurgeon tries really hard to, sorry, my screen just went away. The neurosurgeon tries really hard to be very precise about where they stimulate, but as you can see in the top right, that's 27 different DBS electrodes in 27 different locations. They end up in slightly different spots. So if all these things tell us this common circuit, then we know there's something important and causal about that circuit. And so we started doing that in all the different data sets we could find, 14 data sets, including Vietnam veterans with penetrating brain lesions, a couple hundred strokes all the way from St. Louis to Melbourne, some hemorrhagic strokes in Chicago, 150 TMS targets all the way from Boston to Melbourne, 44 DBS targets all the way from Atlanta to Berlin in patients with major depression, 32 DBS targets in patients with Parkinson's from Berlin, and 25 DBS sites in patients with epilepsy from the Netherlands. And all these patients had depression measured after this focal manipulation. And in short, here's what we found. We take the location of each lesion, for example, put it on a standard MRI brain, look to see what it's supposed to be connected to using a normative connectome database. Now, I should mention, we're not looking at individualized connectivity here. Everybody has slightly different connectivity patterns, but everybody has vastly different lesion locations. So the lesion location greatly overwhelms that inter-individual variability, so we can actually make the assumption that people are more similar than they are different. Of course, if we had individualized connectivity, it would be a little bit better. But then we see if the patient developed depression or not, and we get a map of the connectivity of brain lesions that modify depression. We do that for 5 different lesion data sets. Same thing with TMS. We localize the stimulation sites, see what it's connected to, see if the patient got better, and we get a map of the connectivity of TMS sites that modify depression for 4 different TMS data sets. And the same thing with DBS, stimulation site connectivity, clinical outcome, and a map of the connectivity of DBS sites that relieve depression for 5 different data sets. And so we end up with 14 circuit maps across 14 data sets. And I figure, there's no way this could converge on anything in common, right? I was talking to Mike Fox about this. Again, he's a neurologist, I'm a psychiatrist. He said, check to see what they all have in common. And I said, this is psychiatry. Things don't converge. Things don't replicate, right? But he told me to try it anyway, so we tried it. And sure enough, it showed this. There's the connectivity of lesions that modify depression, the connectivity of TMS sites that modify depression, and the connectivity of deep brain stimulation sites that modify depression. And using spatial spatial correlations with permutation testing, we showed these are more similar than expected by chance. And the permutation testing is permuting one patient's clinical outcome with a different patient's neuroimaging, so we can be sure that the spatial correlations are valid. So in other words, we can potentially use lesions to identify better TMS and DBS targets, at least for depression. And we can use that to actually predict clinical outcomes. So if we generate that map based on 13 data sets and try to predict clinical outcomes in the 14th, we get this. With real stimulation, we can predict it significantly better than with permuted simulations. And this prediction is actually better than what we get with subgenual cingulate as our seed, to answer the previous person's question. So we think this is useful. So, and in fact, we've replicated it a little bit further since then. We found the same thing in 283 patients with multiple sclerosis. Their depression, their lesions are more likely to be connected to this circuit if they end up with depression. So let me summarize what I just told you. TMS, DBS, and brain lesions can all modulate a common brain circuit. And lesions can potentially help us find new TMS or DBS targets, potentially for multiple disorders. So we published this last year in Nature Human Behavior. You're welcome to read more about it if you like, with the help of 26 co-authors, including Robin, who shared data with us. But we originally submitted it to Nature Medicine, and they came back and said, well, you can't call it transdiagnostic if you only tested it in depression. So we said, yeah, that's fair. So they rejected the paper, and we just said, let's try Parkinson's disease, the next biggest data set we could find. So we looked at lesions that cause motor symptoms of Parkinsonism, DBS sites that relieve Parkinsonism, and published TMS clinical trials of Parkinson's disease. And again, these connectivity profiles were more similar than you would expect by chance. So since then, this has also been replicated using tics and Tourette's syndrome and cognitive symptoms of Parkinson's disease. So we were pretty confident this method can help us identify new treatment targets. So based on that, we should be able to extend to other disorders, anything that has lesions. So we looked at the Vietnam head injury study, which is 193 veterans with penetrating TBI and 52 with non-neurological injuries of similar severity. And in fact, TBI actually reduced your risk of PTSD, which sounds strange. But when you think about it a little more carefully or look at it more closely, you find that it was specifically lesions to the amygdala that protected against PTSD. And that kind of makes sense. If you don't have an amygdala, you can't get PTSD, right? But the problem is the amygdala is too deep to reach with TMS. So we actually looked at the connectivity of the lesions that reduced PTSD risk versus the lesions that didn't. And here's what we found. Again, lesion, connectivity, clinical outcome, we get a map of the connectivity of lesions that reduce PTSD risk. And it's connected to what you might expect, the medial prefrontal cortex, amygdala, and hippocampus. We use split half-cross validation to confirm this is real. So you might just say, oh, you're just showing me the connectivity of the amygdala. That's nothing surprising. But in fact, it actually still works even if we drop all the amygdala lesions, we still get the same circuit. So this is actually a network phenomenon, not just an amygdala phenomenon. And it's also true in an independent data set of veterans without brain lesions. They were actually hypo-connected in the same circuit. So this one's not published yet, but we're still working to try to figure out which direction one would want to stimulate this circuit, but we're pretty confident that this one is real. We've also recently done the same thing with addiction. You guys might have seen this paper last week. To the left is the connectivity of stroke lesions that led to remission of nicotine use. And to the right is the connectivity of penetrating lesions that reduce the risk of future alcoholism. And again, these are more similar than chance. I'm going to say that again. If you have a stroke that makes you less likely to smoke, it's connected to the same circuit as a penetrating lesion that makes you less likely to drink. So this is the peak of that circuit. We think it's a good TMS target. This is the field distribution of the TMS coil that was recently FDA approved for nicotine use disorder, and the field distribution of the coil that has recently been shown in a multi-center trial to be effective for alcohol use disorder. So we think this kind of explains why certain things are working. And in fact, based on this target, for future studies, they're going to go a little more anterior. And so this is the paper. You guys are welcome to check it out. It just came out last week in Nature Medicine. And we're taking this further now to look at other disorders. For example, stable personality traits like novelty seeking. We've replicated a circuit for that, which is not yet published. So if you want, either you want to stimulate this circuit if you want to modulate somebody's personality. We've also got one, sorry, clicker is being slow, about mood regulation. So this is the connectivity of lesions that cause depression and lesions that cause mania. So what do mania and depression have in common? We're calling this mood regulation. So right ventrolateral prefrontal cortex, which has been implicated in emotion regulation, and it's easily accessible with TMS because it's on the surface of the brain. So let me summarize again. Lesions can help us find new TMS or DBS targets for personality, PTSD, addiction, etc. And it becomes a question of how to choose the right target for the right patient. Robin was talking about this a lot earlier, but I'm going to summarize again. This was us replicating their work, where we can identify a personalized target based on subgenual connectivity, compare that to the actual target, see how close they are to each other, and use that to predict clinical outcomes. This works. They've already shown it. We've replicated it. So this is one way to personalize. Another way to personalize based on symptoms. We published this 2 years ago, so I'm not going to spend too much time talking about it. People are familiar with it by now, if you were going to read it. So for example, the magenta spots are good targets for what we call anxious somatic symptoms of depression. A different magenta spot also relieves anxious somatic symptoms of depression, whereas the orange spots relieve dysphoric symptoms of depression. This is a whole different talk going into this in detail, but in summary, we can personalize based on connectivity or based on symptoms. So in summary, TMS and DBS can target the same symptom, circuit to treat the same symptom. Lesions can be used to identify TMS and DBS targets. There's a similar circuit in different disorders, including all these, and also now multiple sclerosis. This was based on causal sources of information. We can do it with other disorders like PTSD, addiction, mood regulation, and we can potentially personalize based on symptoms or connectivity. And of course, a lot of people helped make all this happen, including funding agencies, collaborators, people who help share data. I'm happy to take questions, even happier to take postdoc applications. This is my email address, and of course, Twitter is my primary source of scientific communication these days. So, happy to take questions. Thank you.
So thank you very much, Shan. We have 3 minutes left to our session, so I mean it's fair to take a quick question to Shan if we have, and then quickly at Shan, I picked just one. Could you also draw conclusion about the effectiveness of different TMS protocols?
Not based on what we're looking at here, but that's also an important component. Spatial targeting is only one component of optimizing TMS. There's also frequency, intensity, the number of treatments. We can optimize all sorts of things based on patient-specific factors, which we haven't started looking at yet. So somebody needs to look at all these things. We would love to help think together about it.
Thank you.
Thank you. I'll sit down, and I guess we'll take questions for everybody now, right?
Yes. And so we decided to get advantage of this app and to have Victor and Robin to answer to the 2 most rated questions. So the first one is for Victor. Why not reconstructing all fibers, whole brain during pre-processing and then just masking in the ROI in real time?
Yeah, so there are basically two reasons for that. One is usability, which is extremely important in software. The more steps that you add to the user, the less likely is that the user will use it. So we don't need to compute the whole brain. That's basically it. We compute from the ROI that we want in real time, so there's no need for pre-computing. And as far as I know, whole brain tractograms have its own problems, so it's kind of tricky. And the other is the parameter sweep. So basically, if you need to one thing that we do in the real-time tractography for navigation is that we compute at each iteration a lot of different parameters. We compute tracks or streamlines using many different parameters, so we are not restricted to one kind of set of parameters for that, which gives us more flexibility in displaying this information and kind of searching for this kind of, uncertainty of the errors of the parameters that we rise. So these are the main reasons why we don't use whole brain.
Thank you. And then to Robin, how would you define the optimal coil orientation as opposed to the optimal position?
Yeah, I think probably with the prefrontal cortex, you'd probably have to do that based on the E field. Just looking at, what orientation and what angle would optimally activate an individual's brain region in native space. But that question is probably going, trying to relate it back to the motor cortex, where you see that different coil orientations preferentially activate different neuronal subpopulations, like with I waves, I1 wave, I3 waves. It's a bit hard to do that kind of thing, with the prefrontal cortex. It might be a question of trial and error, or looking at TMS EEG or functional nerves, or TMS in the scanner. I'm not sure. Yeah.
Thank you. Or with the new systems that will do it rather smoothly to change the orientation. So with this, our time is finished, and I wish to thank all the speakers and the people that supported me in this great session, at least to me. And overall, I think that we really try to make the point that personalized TMS needs to get help from the other modalities in order to improve like the spatial and temporal structure of what we have to do when hitting the brain with these methods. So thank you very much again. And you can reach us during the next days.
Nice talk, guys.
Thank you, guys.